thought about history books? I mean, really thought about them. Imagine one that was written billions of years ago, telling a story so big it covers every single living thing on this planet. Well, the wild part is, a copy of that book is hidden inside you right now. It's in every plant, every animal, every tiny bacterium, and today, we're going to learn how to read it. Just let that sink in for a second. This isn't some sci-fi concept. It's a real physical record of our entire past, and it's encoded in the very machinery that keeps all of us alive. Now, the language is pretty complex, but the story it tells, it's the greatest detective story of all time, the history of life itself. And the detectives? Well, they're biochemists. So, where is this secret language hiding? It's written in our proteins. You know, we usually think of proteins as these tiny machines that build our bodies and keep things running. And they are. But they're also historical documents. This sequence of their building blocks carries just a massive amount of information about where they and we came from. See, every protein is just a chain of amino acids, and we can represent those as a simple string of letters. This sequence, this primary structure, that's the language our detectives need to learn. And as you're about to see, just by comparing these strings of letters, we can uncover some truly surprising relationships and trace family trees back millions or even billions of years. But think about it. To really, truly understand a language, you can't just read it. You've got to learn how to write it too. For scientists, this meant figuring out how to build proteins from scratch right in the lab. This whole process, called chemical peptide synthesis, was a gigantic leap forward. It's an incredibly careful and, let's be honest, painstaking process. You basically anchor the first amino acid down. Then, one by one, you add the next in the chain, using all these chemical steps to protect and activate each one, just right. You do this over and over and over again until the protein's finally done. It's brilliant chemistry for sure, but it is slow and really complex. And this is where it gets really mind-blowing. To make a tiny protein, just 100 amino acids long, our best lab methods take a few days. You know how long a simple bacterial cell takes? About 5 seconds, with almost perfect accuracy. This incredible efficiency is exactly why studying the protein's nature already made is so powerful. Our tech is amazing, but it just can't hold a candle to biology. Okay, so since nature is obviously the master author here, our detectives focus on reading the texts it's already written. This brings us right to the heart of the investigation, comparing protein sequences from different organisms to hunt for clues about their shared past. And the very first clue these detectives look for are things called homologs. Now, that sounds technical, but the idea is actually super simple. Homologs are just members of the same protein family. Think of them as relatives. They look similar because they both inherited their basic design from a common ancestor way back when. Finding a homolog is kind of like finding two people with the same unusual last name. It definitely suggests a connection. Now, these protein relatives come in two main flavors. First up, you've got orthologs. These are basically the same protein doing the same job, but in totally different species, like the protein that helps build stuff in a human and its counterpart in a bacterium. Then there are paralogs. These are different but related proteins all inside a single species. A good example is the different kinds of hemoglobin that carry oxygen around in our own blood. Now, for tracing that huge grand tree of life, our detectives are mostly interested in the orthologs. All right, so how do you actually compare them? Let's say you've got these two sequences from different bacteria. At first glance, you can kind of see some similar spots, right? Like those strings of G's, but they don't line up perfectly. It's like you're trying to compare two ancient scrolls, but all the lines have shifted and gotten jumbled up. This is where the computers become our secret weapon. Special programs can strategically insert gaps, you can think them there as little dashes, into one of the sequences. And boom, look what happens. All of a sudden, the most similar parts just snap right into perfect alignment. It's exactly like a detective taking two pieces of a torn letter and lining them up so the words finally make sense. Now, this isn't just random guesswork. The computer is running a really careful calculation. It gives a positive score for every identical letter that lines up, but it subtracts the penalty for every gap it has to add. The final alignment you see is the one that gets the highest possible score, which represents the most likely evolutionary connection between them. But hang on, what happens if two letters aren't identical, but they're, really similar? This means we need a smarter approach. We have to get beyond a simple match or no match system. We need something to score the quality of the changes. We need
Basically, a Rosetta Stone for the language of proteins.
This is the million-dollar question. I mean, if an aspartic acid in one protein is swapped for a glutamic acid in another one, that's interesting. They're both negatively charged, they have similar properties. Surely that's got to be scored differently than if it was swapped for something totally different, like a big, bulky phenylalanine, right?
And that's exactly the logic. A conservative change, like that asp to glue, gets a positive score. Why? Because it's an easy swap that probably won't break the protein's function. A non-conservative change, like asp to phe, gets a negative score. Of course, identical matches get high positive scores. And if you get a match on really rare amino acids like cysteine or tryptophan, well, they get the highest scores of all, because a match like that is almost certainly not due to random chance.
This whole logic was put into a system by two scientists, Stephen and Georgia Heinekoff. They studied thousands and thousands of real protein sequences and calculated exactly how often each amino acid was swapped for another. And the result is the Blossom matrix, our Rosetta Stone. It's a master table that gives a specific score for every single possible substitution, and it's all based on real-world evolutionary data. This just shows you perfectly how it works. Take a look. An aspartic acid, which is letter D, matching itself gets a nice high score of 6. An asp matching with a similar glutamic acid, letter E, gets a small positive score of 2. But an asp trying to match with a very different phenylalanine, letter F, that gets a negative score, a -3, which penalizes the alignment. This lets us do incredibly smart and accurate comparisons. So there we have it. We have our method. We can find the relatives, the homologs, we can line them up intelligently using gaps, and we can score them with incredible nuance using the Blossom matrix. With these powerful forensic tools in hand, we are finally ready to solve the case and use this protein language to rebuild the entire history of life. You know how in every great detective story there's a smoking gun? That one piece of evidence that is just absolutely undeniable? Well, in molecular evolution, that smoking gun is something we call a signature sequence.
Let me show you a fantastic real-world example. There's this protein called EF1-alpha, and it has a unique insertion, a little chunk of 12 extra amino acids just stuck in the middle. Now, what's amazing is that this specific insertion is found in all eukaryotes. That's us, plants, fungi, the whole gang, and in a group of ancient microbes called archaebacteria. But here's the kicker. It is completely absent from all other kinds of bacteria. That's not a coincidence. That is a shared fingerprint, undeniable proof that our branch of life and the archaebacteria are more closely related to each other than to common bacteria. Case closed.
And here's the grand finale. This technique isn't just for one or two proteins. By applying these powerful alignment and scoring tools to thousands of different proteins across thousands of species, from the simplest microbe to us, scientists can actually piece together the entire evolutionary tree of life. The history book is finally open for us to read. So, we've gone from a chemistry lab bench right into the heart of our own cells, and we've learned how to read the epic history written inside all of us. We have cracked the code of the past. And that just leaves us with one last incredible question to think about. Now that we can not only read this language, but are learning how to write it, what kind of future are we going to build?